So let's talk about signal transduction proteins, specifically G protein receptors. And so this is what we usually refer to as nature's light switch. It is one of the most common transduction pathways used by any cell type. It's used in everything from taste and smell reception to just standard activation of other secondary messengers within the cell. And just like almost all of our transduction pathways, it's going to begin with this ligand up here at the top, which is going to be some type of external protein, probably a protein. I guess it's not guaranteed a protein, but it's usually a protein. And so it's going to bind to this receptor right here at the top. And remember, this receptor is going to be very, very specific to whatever this ligand is. So think of it as the ligand is the thing that is flicking the switch that is going to cause the activation within the cell. So this little area here, this is my cell membrane, right? And so I illustrated the G protein this way because G proteins are actually kind of unique in their design. They all have this 7 back and forth pathways of alpha helices, stereotypical of G proteins. And then within the cell membrane, there will always be an inactivated protein right down here at the bottom. And so this G protein, this guy is inactivated, and I know he is because he has this GDP attached to it. That is a nucleotide just like ATP, except this guy is very, very specific to G proteins. Also keep in mind that the GDP molecule, when it eventually gets activated, it will be replaced with a GTP molecule. And you might recognize that nucleotide. GTP is the nucleotide that is produced during the Krebs cycle of cellular respiration. So you might have seen that guy pop up occasionally, and it is used pretty exclusively in the G protein pathways for the cell. All right, so again, trying to show the ligand coming in here, binding to the receptor at the top. When that bind occurs, as the picture kind of shows here, this G protein is going to actually bind to the receptor. And so that's why a lot of times we will call this a GPCR, a G protein coupled receptor, because once this ligand binds, it's going to cause this protein to actually move to the membrane protein, and they actually bind together. Once they bind together, the G protein will give up its bound GDP, and it will replace it with a GTP molecule. And again, like you guys are probably aware, GTP molecules, that's a triphosphate, just like ATP. And so this guy right here has a little bit of energy that it can give up, and it can phosphorylate certain things. And so it will actually move in here, and it will replace the GDP, which will activate this entire G protein. And that G protein is then going to do something within the cell. So once that happens, that's going to cause a termination effect to happen at the top of this receptor. So that's going to kick off that ligand. So my little green ball up here goes away. And then the activated G protein will become unbound, and it will move away from the coupled receptor. And so it's going to actually travel oftentimes inside the cell membrane along those phospholipids, and it will travel until it gets to a additional enzyme within the cell membrane. So I've just got this labeled as a fairly generic inactivated enzyme. And so I kind of threw the inactivated in for you guys to show that it'll become activated, right? And so this can be a lot of different things. So if you get a little bit further along in transduction pathways, this can be something like an adenylyl cyclase. And there's literally hundreds of different inactivated enzymes bound to the inside of the receptor. So think of this as just a little generic receptor, right? And so when this G protein is going to come in and it's going to bind, it can kind of do a couple different things. And I'll show you kind of a couple different ways it can do this. But for the most part, it is going to give up its GTP. That is going to bind to an active site here. And once that occurs, that is going to activate this inactivated enzyme. And so I'm showing it here as the GTP being bound. Once that occurs, we're no longer going to call it an inactive enzyme because this guy just got activated, right? And so that's going to cause these little hooked on secondary messengers or maybe scaffold proteins to come free, and they are going to move throughout the cell, and they are going to do whatever needs to be done from activating that light switch. So kind of think of it, whatever this enzyme is, it can have very, very specific jobs to do. Like it might be something that is going to cause additional molecules to begin binding. It might be something that's going to stimulate an electrical response. And so we'll talk about those a little bit later too, but it could cause things like action potentials. It can affect the nervous system. It can stimulate things in the brain, like, tastes and smells that ligand could have induced. But regardless, this GTP molecule, once it becomes bound, it's going to activate this enzyme in some way, shape, or form. I did want to also put in here that because this is a triphosphate, that sometimes this GTP molecule, instead of actually having the entire molecule bind to the active site, sometimes what it actually does is it just phosphorylates the active site, which if you guys know that term, phosphorylate means to take a phosphate off and hook it onto something else, right? And so in this case, the G protein is going to actually activate the site with the phosphate. This phosphate's gone, and then what you're left with is just a GDP. So these two phosphates are what remain, right? So the enzyme's activated over here. The GDP actually goes back to being what it was before. And so I'm showing these internal transaction proteins. They're going to go out and they're going to do other things. My G protein is back to being deactivated. And a lot of times it's going to go back over here and it's just going to kind of hang out and wait for the next time that that G protein coupled receptor binds. Thought I'd also give you guys just a little, I don't know, animated illustration or just simplified illustration perhaps. And so since we always refer to the G protein coupled receptors as a light switch receptor, because it's going to generally cause one thing to initiate the other to occur, I thought I would just kind of give you guys some fun little illustration with it. So same kind of idea here, just give my little ligand buddy here as the receptor. It's going to actually cause this binding to occur. And then I kind of try to draw my G protein in here like a little off light switch, which is going to be the thing we are trying to activate. So same idea, once that receptor binds up here at the top, it is going to actually cause this G protein to flip-flop the GDP like you're flicking a light switch, right? And the GTP comes on. I like to draw my little, I don't know, whatever, his little circle guy here. He's kind of now activating the enzyme, and that is going to cause it to travel along the inside of the cell membrane to where it will then activate that inactive receptor, right? And so he kind of bumps in there. He's going to knock the GTP off. That GTP is going to then activate my receptor. I did, by the way, kind of throw this in as activated because that's already bound up there. And that's going to cause these guys to come bouncing off, right? And so again, just trying to think of it from in my mind, the way this actually is occurring. I always try to think of it as, you know, a little, anthropomorphized, I guess, right? And so, my G protein now, has apparently lost its, GTP wheel. So it's going to replace it with a GDP, which is going to deactivate that specific thing. And my little transaction proteins, really should call them transduction proteins. They're going to go off and they're going to do other things somewhere else in the cell. Once that GDP gets locked back on, though, the car is fixed and this guy can then be repeated, right? And so realistically, this enzyme, it probably will reset after this is all said and done. And so there'll probably be other enzymes inside the cell that will put back whatever was used here, right? And that will cause this to be reset. This guy is already going to be reset. And then that ligand came off of this guy over here. And so everything is really ready to roll again next time around.